Okay, class, this is the last of the animal notes for this week. This is part 4, animal fertilization, development, and germ layers. This is one of the shortest sections of notes, but probably one that you will find that you're least familiar with. There's a lot of new vocabulary in this chapter. And so we'll get started on this, and you have not only this video to watch today, but also another video attached called Just Tubes. That is a really good explanation of what we're going to talk about with notes. So you'll go and watch that when you're finished with this. You can see on the diagram here what we're talking about today is what happens between the time an egg and a sperm meet and create a fertilized egg, all the way until you start to see an organism form and you're actually able to recognize what it is. So that's what we're going to go through today. Okay, so the objectives for today are for you to be able to identify the organ systems that are formed from each germ layer. So we talked about organ systems yesterday. Today we're going to talk about where those organ systems come from. And when we're using the term germ here, we're not talking about bacteria and viruses that might make you sick. We're talking about tissue layers. And then by the end of this lesson, you should be able to listen and explain the steps of fertilization, which you just saw on the previous slide, and how development happens through gastrulation. So again, a lot of new words there. And finally, recognize organisms as acoelomates, pseudocoelomates, and coelomates, deuterostomes and protostomes, and how that relates to an organism's complexity. So hopefully you've been working on your 2 vocabulary booklets this week that are due tomorrow, and those should help you realize what some of these words mean and practice those words. So let's get started with the notes. Again, this is section 4. And as I mentioned before, development begins in animals when a sperm meets an egg and forms a zygote or a fertilized egg, and you can see that in the picture here. As soon as that happens and that zygote forms, those cells start to cleave and divide. Okay, so you can see here, number 1, the zygote or fertilized egg is going to go through cleavage, which are just very rapid mitotic division, so think mitosis. So 1 cell becomes 2, 2 become 4, and so on, until you get a blastula or a hollow ball of cells. I'll give you a minute to copy that. And you're going to see a diagram of this that will help you understand in just a minute as well. And once that blastula forms, that hollow ball of cells, it's going to start to fold inward and create a blastopore. And that blastopore is what is going to eventually become the mouth or the anus of the organism. Remember, we talked about most animals have a mouth and an anus. There are a few that just have one opening, and then some, like sponges, that don't have a digestive tract at all, but those are the exception. Okay, if you look down at the diagram to your right, you see 4 pictures. The top looks very much like the bottom, but if you look closely, the top is labeled protostome, and anus is on the top of this diagram, and the mouth is on the bottom. And deuterostome on the bottom is just the reverse. So in protostomes, like the top picture, it literally means first mouth. So the blastopore, the opening that forms first here, is going to be what forms the mouth of the organism, and then that's going to fold all the way through until the anuses form. So the anuses form second. And this happens in simpler organisms like mollusks, arthropods, and annelids. They form mouth first. In more complex organisms like chordates and echinoderms, this original opening here, the protostome, is what's going to become the anus, and the mouth forms second. So the word deuterostome means second mouth. So here's where we kind of mentioned before, echinoderms, even though they have radial symmetry and no brain, they are more complex in this regard, as in that they develop like humans, like chordates do. Okay, so the process of folding in here to form what's going to be a digestive tract is called gastrulation. So that G-A-S-T-R should be familiar to you, kind of means stomach. So this is how the digestive tract, the stomach, forms. And that folding in is what's going to make the different germ or tissue layers. And those are represented here by the 3 different colors, blue, red, and yellow. And this happens in all organisms except Porifera, except the sponges. Okay. Now, in some organisms like cnidarians, like a jellyfish or an anemone, the development stops here, and they never get a second opening. So I mentioned before, they have a gastrovascular cavity. There's that gastra prefix again, where food goes in one opening, waste get digested. Food goes in one opening, gets digested, and waste come out the same opening. So they have one opening for mouth and anus. So other than sponges, which have no digestive tract, and cnidarians that have one opening, and as well as some flatworms, everything else is going to be a tube shape with a mouth and an anus. Okay, so here's the picture again. Protostome means first mouth, so the opening forms here. That's going to be the mouth, and the anus forms second. And then on the bottom, the original opening here is what is going to be the anus, and the mouth forms second. So the deuterostomes are going to be a little more complex. And again, you can see here the 3 tissue layers that form from that pinching in are blue, red, and yellow. And we'll talk about those a little bit more in just a minute. Okay, so here again, the steps of fertilization: sperm meets egg. That one cell divides into 2, 2 become 4. Eventually you get a morula, and that morula will become a hollow ball of cells called a blastula. The blastula is going to pinch in through gastrulation, and that's where you get 3 germ layers. The ectoderm on the outside, meso in the middle, and endo on the inside. Again, with cnidarians, there's only going to be 2 layers because it stops right here. And then sponges have no germ layers because they don't have true tissues. Remember, their cells act individually. Okay, and then you get that folding in. So here's a picture of all of it: the zygote, the cleavage, you get the hollow ball of cells, the gastrulation, and then the tissue layer starting to form. And here you can see there's only 2 tissue layers because there's only one opening. Okay, so here are the 3 tissue layers that I mentioned. Organisms like cnidarians that only have 2 layers of tissue, the ectoderm and the endoderm, they're diploblastic, the prefix di meaning 2. Animals that have bilateral symmetry all have 3 layers of tissue, and they're triploblastic. Okay, so the diploblastic are going to be cnidarians. Triploblastic is going to be all the worms, arthropods, mollusks, echinoderms, and vertebrates. So basically everything except cnidarians and sponges. The layers of tissue are probably pretty easy to remember because of the prefixes, ecto being the exterior layer of tissue, meso in the middle, and endo on the inside. And the reason we care about those is because the organs that come from those are going to depend on the location. So the outside organs that come from the ectoderm is going to be the skin, the nervous system, and your sensory organs, all the things on the outside of your body that sense your environment, protect your body on the outside. Underneath that, the middle layer is what comes from that is your skeleton, muscles, excretory system, which is your kidneys. So if you've ever been hit in the back, hitting the kidney, that's very painful. That's because those are pretty close to the outside of your body. Not as internal as the endoderm organs. And that interior layer is what is going to turn into the digestive system of the organism, the respiratory system, and other things on the very inside of your body. Okay, so here's just a representation that shows you what comes from the ectoderm, nervous system, brain, skin. Underneath that, muscle, bone, kidneys from the mesoderm, and the very internal organs, digestive tract, comes from the endoderm. Okay, and those are just form different systems depending on what tissue layer it is. Okay, and then finally, 3 new words here. All have a root that's similar, a coelom. The word looks like it would be pronounced colum, but it's pronounced coelom. And a coelom is just a true body cavity. You may have heard the word body cavity before if you've watched any medical shows. So if you put the prefix a before coelom, it means without. So flatworms would be like this first picture here. They have no space around their gut or digestive tract. They are flat, literally flat. A means without a coelom. Roundworms, pseudocoelomates, pseudo meaning false, like the pseudopods, the false feet of amoebas. They do have space around their digestive tract, but it's not lined. So it's a false coelom. And then everything else that's more complicated than that, mollusks, arthropods, echinoderms, and vertebrates, have a true coelom. They are coelomates. So they have the space here around their gut, and you can see that it's lined on both sides by tissue. So it's a true coelom. So flatworms would be like this picture, roundworms would be like this picture, and then everything else more complicated, like segmented worms, arthropods, mollusks, are all like this picture. And remember, we don't see cnidarians here because they are not like tubes. They only have one opening, and sponges have no tissues. So acoelomate, pseudocoelomate, and coelomate. Okay, this is the video that you're going to need to go watch that's attached to this assignment. It will help you understand all of that, and you're going to watch a salamander develop in tomorrow's lesson.